invite Nilesh, because just to give us more understanding about biosimilars, that what exactly, because there are 2 3 terms which are being floated and highlighted, and sometimes it is different for a lot of people to differentiate these terms. Thank you, Dr. Tiwari. You can, thanks for all your help.
Thank you, sir. I would like to thank first Ashish sir for inviting me, and it's a privilege to be a part of this such a wonderful panel. So these are my disclosures. I am going to talk about biologics, biosimilars, and biobetters. What do they mean? So this is the public perception as what we were talking about. What is biosimilar? It is just a cheap copy of the innovator or the main thing. That's what most of the sad to say, because innovator companies also have created the conception when the biosimilar came in, not in ophthalmology, but in other fields. This was a gimmick that was played out, but then as soon as our understanding started, we got to know what it actually is. It actually holds the same basic structure. It has the same passion. It has the same primary structure. So all the amino acid sequence in innovator and biosimilars are exactly similar. It's just during the post-translational modification when the protein is formed, and then when it is getting folded, it's a secondary structure, tertiary structure, then the post-translational modification, if it is having a pegtail or something, there the difference lies. And that difference is not that much. It has the similar efficacy and similar safety. That is the baseline. So each drug, even if it is not having the same the quaternary structure, what we call, it has the same primary action site, and it has the same secondary action site, and it has the same efficacy and the same safety. So by definition, there is no standard definition across all of these medical agencies, but the crux what we could find was highly similar to approved biologics, notwithstanding the minor differences in clinically inactive components having similar safety, purity, and potency. So how are they manufactured? So the manufacturing actually...
Can I just interrupt, Nilesh? Go back to the previous slide. I can interrupt him. He was my student. So this is, a very important part, that you cannot be same. A biosimilar cannot be same as innovator, and that's where somewhere negative perception starts. That these are copy, they are not similar. But now it's important to understand, if you take a batch of Lucentis from Europe, if you take a batch of Lucentis from US, they would be different. Correct me, Alok, if I'm wrong. So because biologics are, kind of molecules which is very difficult that you really match each and everything. They are not generic, they are not chemical formula, because they go through the living organisms, and living organisms, there are few changes in terms of buffer, chemical are there. So that's what it defines, that clinically inactive component. So those are the components which do not matter in terms of the efficacy and the safety of that particular drug. So always remember, even 2 different Lucentis are also different. So it's not something that only biosimilars are different little bit, but they are all into the inactive component. So that negative perception should not come. Thank you. Sorry to interrupt.
No worries. So the workflow starts when the patent, actually it has to start when the patent expires, but as we know, R&D starts much earlier. So there is a reverse engineering on the proprietary molecule. So there is a lot of analytical, there is spectroscopy and more analytical thing that goes, so that they can find out what the actual primary amino acid sequence is, because that is a proprietary thing. The innovator is never going to tell, even if the patent expires, that doesn't come into public domain. So the basic amino sequence is interpreted and kept same. Then the secondary, tertiary, and quaternary structures, they might be different. So that the thing is to understand is the cell line from where the molecule is being created. It has to be similar. It has to be, if it is E. coli, it has to be E. coli. But exactly the same cell line basically is not used and should not be used. The cell line for production might be different, and the purification process might be different. And this is a point that we will ponder about when we are talking about Biobetters, because the new technology, the biosimilars which are getting manufactured right now, are 20 years ahead of their innovators. There is more purification, there is more better cell lines that are being used to protect. So Biobetters, when we come to, we will know why this is important. So then the workflow, Ashish sir will talk about. This is what I say. Let the master show how it is done, how they are regulated and how they are studied. So why do we need them? There is what device I was telling about. There is a direct discount that is happening, and then there is an indirect benefit from the lesser disease burden. So the patient who are getting treated, they are more economically active, and they are contributing more into the society. But what is inapparent is the wider availability playing to demand and supply. So when there is more supply, the demand, when there is more demand and supply is more, the prices come down. So that's what is happening. We are seeing with innovators, they are now starting, they have started patient benefits, which was not available earlier. But as soon as the biosimilars started coming in, there is the patient benefit came, and even in the biosimilar now the prices are coming down. And here one point I want to tell about our pharma partners: please stop these schemes, 10 pe do free, 20 pe 5 free, because anyway you are going to give that amount to everybody. But if you are going to get the baseline below, so even if it is like 7,000, 8,000, 10,000, whatever the baseline you are going to create, if it is flat, the smaller practitioners who are going to inject one injection per month, they will also afford to buy that and inject that. So please stop these schemes and give a flat rate. So more and more people are going to use it. So this is a sincere request. And is it all rosy? Is there any side effect from biosimilar? You can see the graph that is about antibiotics. We are right now into that ascending phase. There is innovation that is going to happen, and then there is a plateau, and then now for last 30 years we don't have a new antibiotic. We are in the ascending phase of this biologics. We are having every 2-3 years we are going to have new molecules, but soon we are going to plateau. And there my humble request is, don't stop innovation, because as you can see on the other chart, that is the generic chart, the amount of economics that is into play for this generic. So as more and more generics are being produced, the innovation keeps coming down. So as more and more biosimilar will keep coming, the innovation should not stop. That is one other sincere request from my side. So what are Biobetter? Dhoni is the Biobetter of Kapil Dev. Do you agree or not? There was already one World Cup at that time. Now he has gone ICC trophy. He has got T20. Similarly, some can say Kapil ke time mein there was no T20, there was no ICC Champions Trophy. Exactly the point. We have now better manufacturing capacity. We have now better purity. So that's why that is going to be a biobetter. It is but natural that they are going to be biobetters. Biobetters, there is no standard definition, but it has similar primary structure as biosimilar, but it has greater potency. Either it can have greater potency, it can have greater shelf life, it can have greater half-life. Anything can happen, but in one or more than one aspect, there will have a better, it will be better over the biosimilar. So these are the two potential very great molecules having very great efficacy, but they are marred. Abicipar pegol, it couldn't come into market because the IOI rate was at 8%, and brolucizumab, after being launched, is not being used that much because of the high IOI rate. So these in brolucizumab, it's very simple. You add a pegtail, you reduce the antigen component, like what we are injecting. If you reduce, there is high chance that the IOI rate will come down, because it's just what we propose. It is a type 3 reaction. The less antigen you inject, the less antibodies it will interact with, and lesser IOI. So brolucizumab is one where biobetters will help, and abicipar pegol, because of the more purity that requires from E. coli components, so it will also benefit. But why there is no biobetter? Because it is considered to be a new drug. It won't have a abbreviated licensing protocol as biosimilar. So again, the entire R&D. So even if the initial cost of developing a molecule comes down, the trial cost is same, and that's why we need a regulatory change to get biosimilars. Each of the company, personal experience, Rani has no financial interest, but Rani has better drying capacity than the innovator molecule. Razumab also was better over the proprietary, but Rani has, because it is more recent, it has even better drying capacity. But no company, neither Lupin will claim that, neither Intas will claim that, because for claiming, they need to go through that entire thing. So it is only whispers. So there is required a regulatory change to bring Biobetters. Thank you.
So thank you, Nilesh. I mean, that was very well highlighted. So Biobetter, I'll just put a point on Biobetter that actually, if you really see in a simple sense to understand biosimilar Biobetter is actually Lucentis was a Biobetter of Avastin, because Avastin was a full-length molecule with FC portion, with FAB arms, and what was done that it was seen that it works, and so it was just modified in a way so that molecule size decreases, and you cut down FC portion. So ultimately, that's how the Biobetter happens. But what happened? Lucentis came as a innovator molecule. It was essentially a Biobetter. So Biobetters means have a long road to really have that regulatory hurdles. Means we understands a little bit. I think I would like Alok to comment on this. Means where, because he is been into industry for quite some time. So Biobetter, what do you see, Alok? Means Alok is he's Dr. Alok basically. He's from Intas, so he understands. I mean, I keep taking his guidance for many of our papers. So means he is the right person to comment on that.
Biobetter is always an interesting thing, which is like somewhere you need a complete big innovation, somewhere frugal innovation works. There will be no ways where we can see what is better or what is the way to follow. But I think even regulators now have defined a particular pathway for biobetters, and because of a gap in understanding how much innovation is real innovation, they've kept it more like a innovator product or a complete new product which is to be which you need to discover. Not probably from a biobetter example, and you shared a fantastic example. Lucentis is nothing but a biobetter of bevacizumab. We had certain histories with biobetter which probably didn't work when the numbers were expanded. So typically the trials, you see sometimes a very minor difference in a smaller trial, which when you replicate on a higher population size, that just disappears somewhere. And it's not always a statistical error. But just that you include more people, that biobetter most of time becomes to be a biosimilar. So a classical example, when Dr. Nilesh shared the drying potential, absolutely we make a comment of drying potential on our observations, which are the newest first. But when we put it to the numbers, and I can vouch with all the trials, Rani-Eyes trial is published, Razumab trial is published, you see CFT thickness same. So that's where biobetters run in trouble.
The trial are designed for non-inferiority. If we have a statistically superior
Yes.
trial design, this might be different.
So two things into it. They are designed for non-inferiority to reduce the population size involved. Rest, non-inferiority and superiority trials, if you design, typically you are only the subject size changes. Apart, of course, from your primary endpoints and statistical plan. But that's where that biobetters doesn't have yet been established as a fact, because you never know if you put it to the mass which way that biobetter would work actually as an innovator, or that small difference seen might actually become nullified in the efficacy or safety when we see, and that's where the problem is with biobetters.
True, true. So means, but I'm glad to hear that some path is being given now.